I'm excited to share with you today some very new ideas and new data that has never before been shared with the public in the sphere of optogenetic biomanufacturing, which is a new field that my team and I have been pioneering. And yeah, hope you enjoy it. So I have 20 minutes with you today, and in these 20 minutes, I want to give you a glimpse of what is coming down the line in biomanufacturing. And there's really three main things that I want to cover. The first is what's happening in biomanufacturing today and what the implications of that is to current biotherapeutics. The second is introducing optogenetic biomanufacturing and explaining what that is and how it works. And the third is explaining how people can use it and what that means for your businesses if you choose to. So I think it is no surprise to anyone in this audience that when we look at the data on biotherapeutics, it reveals a very interesting trend, which is that between now and 2035, the pipelines are growing, but the distribution of which assets are growing is kind of unexpected. It is not the typical mAbs. The growth engine of biopharma is coming from bispecifics and trispecifics and multispecifics and fusion proteins. And what's super interesting is that we don't have good ways of manufacturing these drugs right now. They can get manufactured, but they tend to end up with titers of half a gram per liter, 1 gram per liter. If you're lucky, 2 or 3 grams per liter. And this is a problem because if we have 80% of the pipelines going to be with drugs that can only get made at sub 3 grams per liter, that's going to be a big issue for the industry, and it's going to be a big issue for patients who need these drugs. So what that's going to lead to is an industry where you have a big COGS problem for the growth engine and some amount of a COGS problem for mAbs relative to small molecules. But there's this opportunity here in the top left-hand corner, and the opportunity is if you can create tools that can make these drugs with extremely high precision, that can overcome the toxicity issues, that can overcome the need for chain ratio control, that can overcome the need for some of them having short half-lives, then you can really unlock this growth engine. So this is where optogenetics comes in. So the idea with optogenetics is that you have light-sensitive proteins, and these light-sensitive proteins already exist. They've existed for billions of years. And what they do is they have one shape in the dark, and they have a different shape in the light. And many of them exist, and they exist in many different wavelength activations. So for example, some of them can be activated by blue light, some of them by red light, some of them by far red light. And what we do is we take these proteins and we tether them to various different targets inside the cell. And that allows us to control the activity of those targets using light. So not only switch them on and off, but dynamically tune them. And this allows us to control any process inside of the cell using light. And it's pretty crazy. Like, as a scientist, I've worked a lot with traditional tools to control biology, which are all indirect, changing the pH, changing the media, changing the temperature. And it's a real paradigm shift because all of those things perturb the entire system. So what we're talking about here is having tools that can perturb a single protein inside of the cell without perturbing the rest of the system. And doing that dynamically is a profound shift. So, to give you an idea of how this could work, it's not just, separating out growth from production, it's finding a certain pattern of production that is best suited for the molecule that you want. So, for example, if you have a toxic protein, you can control the transcription with light and produce it at the dose where it never exceeds the toxicity thresholds. Or for example, if you have a bispecific or a trispecific, you can have the heavy chain and the light chain under different colors of light, and then be able to pulse the light to control the stoichiometry so you don't get the chain mispairing issues. So just to show you some data here. So on the left-hand side, there is a bunch of clones that were made with optogenetics, and on the right-hand side, a bunch of clones that were made with conventional tools. And usually with conventional tools, which Most people use constitutive promoters, which means that they're always on. What you have is a trade-off between high production and fast growth. Because if you have really high production, that tends to come at the cost of not being able to grow as fast. So those clones get outcompeted in your population. And conversely, if you have low production, then you have a low doubling time. Because if you have low production, then you're not putting a lot of stress on the cells, and then that allows those cells to grow faster. So with a traditional process, you frequently end up with clones that can make a lot of protein, but don't grow very fast, or grow very fast and don't make a lot of protein. With optogenetics, you can have the best of both worlds, and that's what you're seeing here. So on the y-axis is protein production, on the x-axis is a doubling time, and what you can see is you can have clones that both have higher production and have lower doubling times. And this is very beneficial because you can split out the production phase and the growth phase, and then when they're in the production phase, you can pulse the light to get the production to exactly what you want. So just to give you guys an idea of what we've done in mAbs just in the last 12 months, we're currently at 13.5 grams per liter on fed batch for IgG1 mAbs, which is pretty good. You know, the industry is currently at, for fed batch, somewhere between 7 to 10 grams per liter. We don't quite know what the ceiling is with optogenetic biomanufacturing in terms of mAb titers, but we know that it is not 13.5 grams per liter. We think it is probably north of 20 grams per liter, and that is our goal for the end of this year. And it goes beyond titers. So this is some very exciting data that we've been generating on controlling glycosylation. So remember when I said that any protein inside the cell can be controlled with light. So here we're controlling proteins that are enzymes that control the addition of sugar moieties to the amino acid chain. And what we've shown is that we can tune these enzymes using light to very precisely control when a certain sugar moiety gets added and where the sugar moiety gets added. And we've done this for galactosylation, and we've done this for silation. And one of the implications here is that if you can control the sugar moieties and you're an originator, this allows you to tune your mechanism of action. But if you're a biosimilar maker, this allows you to very precisely match the glycosylation profile of the drug that you're trying to copy and have that be guaranteed, because you can have extremely precise, fine-tunable glycosylation, which is what you're seeing on the right-hand side here. So we believe that this will lead to a whole new era of biosimilars, which we're calling bioidenticals. And yeah, people have been very excited about this so far. So just to give a brief summary of achievements in 2025 for mAbs, we were over 2x absolute titer improvements over gold standard sequences. For difficult to produce proteins, we were also over 2x. For toxic proteins, we were over 3x. For glycosylation, we're doing first-in-class control. And this year, we're doing a big push on bi, tri, and multi-specifics and the chain ratio control. So how do you actually use any of this? Our goal is to make this available to everyone. So if you are an innovator, or if you are a CDMO, or if you're a big pharma company and you want to do this in-house, we want to make it super easy for you to do this. So we offer the full stack of solutions. So we can do cell line development. We work with manufacturers to enable them to do optogenetics. So we give them the hardware and the software they need. We do white glove customer service. So you don't need to know anything about optogenetics. This is just like, we have a team of engineers that goes to you and sets this up if you want to do it in-house, or we tech transfer to CDMOs, and there's multiple different CDMO options for our customers. So in order to enable that, we have a suite of hardware that is plug and play with the existing infrastructure that allows you to actually shine the light on the cells. This isn't sold separately, but it is part of the service, and it doesn't require you to have any new capacity build out. So this is all inserted into infrastructure that already exists. Either your infrastructure, if you have infrastructure, or if you don't, then this would be at a CDMO waiting for you as your asset comes into them. And if we zoom out a little bit, really what's happening is if you have cells under the control of light, and light is a digital input. Essentially what you're doing is digitizing cells, and you're having a layer where, because you now have a digital input controlling a biological machine, you now have a system where machines can start learning how to control those cells. And so we have a team that does this, and what they've been doing is experimenting with different light patterns to see, can we train a machine to learn what the optimal light patterns will be to get to a certain outcome. And that outcome could be, a certain mAb at a certain titer, or a virus, or a bispecific, or a trispecific. And we have demonstrated that this is possible. So let me walk you through this. So there's a dotted line here that I hope you can see, which is the set point. This is the target that we set for this experiment. And it's basically this graduation of protein production that we were trying to do. And in green is what human experts thought would be good light patterns to use to hit these targets. And in blue is what our algorithm chose to do. And the algorithm doesn't make any decisions in advance. So it's just taking all of the sensor data from the bioreactors, and it processes that data, and then it makes predictions on what is the best light pattern going to be. And what you can see is when you look at the decisions the algorithm were making, it's kind of all over the place. It's going up and down, and it looks erratic. But if you look at the output, it is far better than the humans at hitting the set point. The humans look way more stable when you're looking at the decisions they're making here in green. But when you look at the output, they're not as good as the algorithm. And the bottom panel is showing that the viability is the same, regardless of whether it is human experts or the algorithm. So I believe that this is the early demonstration that driverless bioreactors are possible, that we can have algorithms that will live inside of your bioreactor that will make decisions for you, and that will be a very good thing. It's like, imagine if you had bioprocess control where you have the cumulative knowledge of hundreds or thousands of bioprocess engineers that work 7 days a week, 24 hours a day. They never have a bad day. They're never going through a divorce. They're always there. They always know what to do. And that's essentially what we're building here. And we have a rich pipeline that is across all different aspects of biopharma. And I just want to give one particular case study, which is a real company. And it's a company that's using optogenetic biomanufacturing. And this company was stuck. So they had raised a ton of money, in the hundreds of millions, and they have a very promising drug. And they'd gone to 4 different CDMOs to try and get this drug made. And none of those CDMOs could get them above 0.4 grams per liter, which is the territory where if you throw enough money at this, you can still do your clinical trials, but it is not the territory where you have a profitable business. Now with Prolific, for these kinds of molecules, what we typically see today is 2 to 3x yield advantages. We think we can get a lot bigger than that long term, but 2 to 3x is what we can see today for this type of molecule. And that may not sound like a lot, but if you actually put it into the context of this company, that means you can save over $100 million per year for this company, and that means that they can progress their molecule into phase 3. That means that they can have a viable business at the end of it. So the lesson here is that if you have an asset and it's at the 0.5, 1 gram per liter, 1.5 gram per liter, where you're worrying about whether you have a real business here, not because the drug doesn't work, but because the manufacturing infrastructure currently isn't good enough, don't let yourself get into this position. There are now tools available to help you. You can come and talk to us. We have various people from the Prolific Machines team here, and we'd love to help you. You no longer need to put up with low titers for your complex molecules. And yeah, I'll end with sharing something that today may seem controversial, but I think will become obvious over the next few years and decades, which is that I think that all biomanufacturing will eventually be optogenetic. And the reason I believe that is because optogenetics allows you to have the lowest possible cost and the highest possible control and precision and quality at the same time. And historically, when technologies have coupled highest possible quality and precision with lowest possible cost, they tend to get adopted. And what I think you'll see is that this will be slow. It will happen over the course of a decade potentially multiple decades. And it will start with the assets that need it the most, these complex protein therapeutics that can't get made anywhere else. But then you'll see a slow spread. So it will go into mAbs, where we're already seeing really compelling data. Then it will go into viruses. Then it will go into vaccines. Then it will go into regenerative medicine. And then eventually it'll go into lots of other industries, even beyond pharma. And so my email is dennis@prolific-machines.com. If you're listening online and you want to learn more, please shoot me a note. We'd love to talk to you and share more of the data with you. And if you're here in person, I'll be in this room for at least another 30 minutes. I'll come sit over here after this talk. If you are an asset owner and you have this problem, come and talk to us. If you are a CDMO and you want to future-proof your manufacturing, come and talk to us. If you're a big pharma company and you don't want to use CDMOs, but you have some of these molecules and you want to do the in-house manufacturing route, come and talk to us. We can help you as well. Thank you very much for your time. Really appreciate it. I'll be down here. Bye.